really two main diseases that are joined under this umbrella term. For one, there is chronic bronchitis in some of the patients. It's a narrowing of the airways. This can be due to structural changes, so more mucus that is obstructing the airflow, but there's also a lot of inflammation going on in the airways. And so the patients mainly suffer from a problem exhaling the air. So if you want to imagine what it feels like, then you would inhale and try to exhale, but not exhale fully, and then take a new breath. So there is more air that remains in the lungs, and so they're breathing from a higher basal volume, and so also smaller breaths. Now, another part of the problem in COPD is emphysema in the lower image, and that is that actually the gas exchange area, the alveoli, they are damaged. And this can be due to proteolytic activity of a lot of enzymes. And so you not only have an obstruction limiting the amount of air patients can exhale, but there is also a limited surface area for gas exchange available. So there's also patients suffering from lower oxygen levels that are distributed to the tissues. Now, the main cause of chronic obstructive pulmonary disease is cigarette smoking, but not every smoker will eventually develop COPD, just like not every smoker will develop lung cancer. So it's about 25% of the smokers who develop COPD, so you can also pose that 75% will never develop COPD. And therefore, people think that genetic predisposition is one of the main causes for developing or not developing the disease. So if we think of oxidants in the context of COPD, there's a couple of different sources. For one is the cigarette itself. The smoke that people inhale contains a lot of different oxidants, and I've listed some on this slide. On the other hand, COPD is a chronic inflammatory disease, and the main inflammatory cell in the lung of COPD patients is the neutrophil. Now, the main enzyme in neutrophils is myeloperoxidase, MPO, which catalyzes a lot of the oxidant formation and also secondary oxidative products. And what would be the reason for the inflammatory cells to produce oxidants? That is to attack particles that come in. And in the COPD patients, this of course is the smoke, but it can also be bacteria or viruses. A third source of oxidants in the lungs is epithelial cells themselves. So the lining of your lung, the normal barrier function cells, they also can produce oxidants. For one, there's nitric oxide synthases, but other enzyme systems that produce superoxide as well as hydrogen peroxide are present in these cells. So the oxidant production is not only limited to inflammatory cells, it also can occur in the resident cells of the lungs. And this has not been discovered all that long ago, so there's a lot of research going on as to why also resident cells produce low levels of oxidants. So in general, there is an imbalance between the level of oxidants and the antioxidants that should protect the body from the oxidant insults. In COPD, a number of oxidants have been found to be increased, such as hydrogen peroxide as well as nitric oxide. But there's also lower levels of certain antioxidants, and the main antioxidant in the lungs is glutathione, so a simple tripeptide molecule that can scavenge oxidants. There's also a lot of vitamins present in the lung, and also enzyme systems such as superoxide dismutase. Now, so normally when your cell, your body encounters oxidative stress, it will try to protect itself by increasing the antioxidant defenses. And the main transcription factor that is involved in inducing the transcription of genes that lead to antioxidant enzymes is this very complicated name, nuclear factor erythroid 2 related factor 2. So it's a complicated name, and erythroid, it was first discovered in red blood cells, but it's present in most all cells. So what normally happens is that when the cell encounters oxidative stress, the inhibitor of NRF2, which is KEAP1, will be degraded so that the transcription factor can go to the nucleus to perform its function. How does it perform its function? It will recognize, it will specifically bind to certain promoter elements in the genes that transcribe antioxidant genes. And these regions are called antioxidant response elements. So it's a basic consensus sequence that is complementary to NRF2, and binding of the NRF2 to the promoter element the antioxidant genes will lead to more transcription of the antioxidant genes. And these are, for instance, heme oxygenase, an important antioxidant, but also a lot of the enzymes that code for glutathione synthesis. So part of the problem in COPD is that this antioxidant defense system is not adequately activated in response to oxidative stress. And there's multiple ways that this is malfunctioning in COPD. There are certain SNPs, so single nucleotide polymorphisms in, for instance, NRF2 itself. But there's also a lack of degradation of KEAP1, so that you don't get the liberation of NRF2 to go to the nucleus. So oxidative stress in this schematic is part of the amplifying processes in COPD. And it's connected to a lot of the cellular processes that are malfunctioning in COPD, such as, for instance, apoptosis, inflammation, repair, and senescence. But I guess the main question that we're trying to address is how does oxidative stress really contribute to all these cellular processes going wrong, to the pathology and to the clinical outcomes? So how do oxidants influence these processes? That's by oxidizing the macromolecules in the cell. That's not only DNA, it's also proteins, sugars, and lipids that are being oxidized. The oxidation leads to a malfunction or a not proper functioning or alternative function of these macromolecules, which eventually leads to disturbed cellular processes and pathology. So in that context, we typically think of oxidants as damage, but oxidants can also function as signaling molecules. And one of the things to keep in mind with oxidants is that they will not travel from where they're produced to where they have an effect, because they're so highly reactive. They will react with whatever is in the area of where they're being produced. So in that respect, if you target the production of the oxidant to a certain area, that will determine what target it affects. So if it is produced in the nucleus, it can have an effect on a nuclear protein or DNA. If it's produced in the membrane, it can have more of an effect on membrane proteins or lipids. So if we consider the damage versus the signaling, damage is typically irreversible. So if a protein, DNA, is damaged, it will remain oxidized, it cannot be repaired. And therefore it's also not regulatable, because you cannot reverse the oxidation and restore function. It's typically also non-specific, and it does regulate function. Whereas if we consider oxidants as signaling molecules, it, by, for instance, being formed somewhere very specifically, can target specific targets. It's also a reversible oxidation, so there are enzyme systems that can reverse the oxidation and restore the function. It can also be very specific, so it can oxidize a certain amino acid and not another. And it also will regulate function. So coming to an example of a redox-dependent post-translational modification, so right side of the slide. One of the main cytokines in COPD is tumor necrosis factor alpha. It's not only an inflammatory cytokine, it can also induce apoptosis. And this is a signaling pathway that normally is activated when TNF binds its receptor. So the main transcription factors that are activated upon TNF binding are NF-kappa B, which leads to survival, proliferation, and inflammation. And on the other hand, you also have activation of AP1, which leads to apoptosis, differentiation, and inflammation. This is a simplified schematic, so TNF binds its receptor. You get recruitment of adapter proteins. This will activate the inhibitor kappa B kinase complex, which beta subunit is really important. You will get phosphorylation of inhibitory kappa B alphas. They will be degraded. So just like with NRF2, the actual transcription factor complex can move to the nucleus and lead to transcription of genes, cytokines, adhesion molecules, amplifying inflammation. What we questioned in previous research was, what if you have a cytokine in the context of oxidative stress? So we incubated cells with TNF-alpha, but in combination with either the oxidant hydrogen peroxide, or we had the cells produce more oxidants themselves. which is of course much more physiological than adding hydrogen peroxide from a bottle. And so therefore we added this NOx system, so the epithelial oxidant source and its co-activators. And what we found is that the co-incubation with oxidants and TNF led to the oxidation of inhibitory kappa B kinase beta at a certain cysteine residue, which limited the phosphorylation of I-kappa B alpha, so there would not be translocation of NF-kappa B to the nucleus, and there was less inflammatory signaling. So this is an example of an experiment in which we proved this. So we looked at the activation of oxidant production with NOx and the co-activators, and in the top panel we looked at IKK activity. So you see, compared to pcDNA, which is the control, the NOx system would limit IKK activity. And in the bottom panel, we transfected in an IKK version with a cysteine mutant. So we deleted one of the cysteines at the critical active site into an alanine. And this cannot be oxidized. So the alanine is not oxidizable, the cysteine is. So if you just change this one amino acid in IKK beta, then the oxidant can no longer inhibit the activity of the kinase. On the other side of the slide, we looked at the actual oxidation of IKK beta. So that's IKK GSH. So it's actually GSH that is being coupled to the kinase. Here again, with hydrogen peroxide, you see an increased band thickness, so there is more oxidized IKK. And if we let the cells recover, so we wash away the hydrogen peroxide, let the cells recover for 20 minutes, then the oxidation is restored by itself. So the cells have a system to reverse this inhibition, to reverse this oxidation. How do the cells reverse this oxidation? Well, I tried to draw the parallel with phosphorylation. So a protein can be oxidized by glutathione to form the protein GSH. And there are enzyme systems that can reverse this binding, and these are glutaredoxins. So if you look at phosphorylation, it's a kinase that attaches phosphate of ATP to the protein, which will modulate the activity. And there's enzymes, phosphatases, that can remove the phosphate from the protein and restore the activity. So in this specific oxidation, the binding of glutathione, the oxidant is sufficient to make the coupling between the protein and glutathione. And there are enzyme systems, such as the phosphatases, that can remove the glutathione and make the protein reduced again. So in our system with inhibition by oxidants, we also wanted to see if these glutaredoxins are really involved in restoring the activity naturally in the cell. So we overexpressed glutaredoxin, we gave the cells extra glutaredoxin, and then we see that the oxidation is reversed and normal signaling by TNF can take place. So here again we see TNF activates IKK activity. If you combine TNF with oxidant, hydrogen peroxide, you limit IKK activity. On the other side of the slide, you see hydrogen peroxide induces the oxidation of IKK. If we put in extra glutaredoxin, then we see that the hydrogen peroxide is no longer capable of inhibiting IKK. And on the other side of the slide, you see that extra glutaredoxin also prevents the oxidation of the kinase. We also did the converse. We knocked down glutaredoxin expression with siRNA approach. And in the lower part of the slide, you see that if you have less glutaredoxin in the cells, then TNF is less capable of activating the kinase. And hydrogen peroxide is more capable of inhibition. And on the other side of the slide, you see that hydrogen peroxide can induce more oxidation of IKK. Now, does this really matter? We also looked at NF-kappa B itself. These are pictures of epithelial cells that were isolated in the top from wild-type mice. So we just take the trachea and we isolate the cells of the trachea from mice that are either wild-type or do not have glutaredoxin by transgenic approaches. And so the knockout cells are in the bottom. In red is NF-kappa B stain. So normally if you treat the cells with LPS, then the red stain will move to the nucleus. The nucleus is stained in green. So you get the green and red overlap turning into yellow NF-kappa B nuclear staining. When we do this experiment in cells from glutaredoxin knockout mouse, you do not see the green staining, so it's indicating that the NF-kappa B is not moving to the nucleus. Then we looked at one of the very important cytokines involved in neutrophil chemoattractant, so very important in COPD. And you see again that wild-type cells that have been treated with LPS, you get a nice response, so a high level of IL-8 or KC production from these cells. If you do the same experiment using cells from GRX knockout mouse, then you do not see this enhanced KC or IL-8 level, and so we expect then there would also be less neutrophil influx. So the glutaredoxin 1 levels seem to determine the extent to which NF-kappa B can be activated, or that oxidants can inhibit this pathway. So more glutaredoxin led to more NF-kappa B, whereas less glutaredoxin to less NF-kappa B. But this was all just using cell culture. And you can prove a lot in cell culture, more than you can prove in a clinical setting, but does it really matter in a clinical setting? So to go from the in vitro experiment to the clinical setting, we first used an animal model of COPD. And an animal model of COPD is like it is caused in humans. You expose mice to cigarette smoke. Now, some people might think that is awful, and the first couple of days the mice also think it's awful to be inhaling cigarette smoke. But people do see that the mice, just as humans, get attached to the nicotine. And so they know when the cigarette smoke is coming, and they're really happy to receive it after a couple of days, a couple of weeks. So in patients, you start smoking, I don't know, some people very young, some people later on, but the disease often only develops when people are in their 60s. So a mouse, it's also not 2 days of cigarette smoke and they will develop COPD-like symptoms. To really develop COPD in a mouse, so the destruction of the lung tissue, it takes 6 months. So these are really, really long and expensive experiments. So we started by looking just at 4 weeks, which you already see the characteristic inflammation. And because in vitro, our main data were directed towards inflammation, we also looked at the inflammation induced by cigarette smoke. So what we did is we used wild-type mice and mice that were deficient in glutaredoxin 1. They received the smoke of 5 cigarettes 4 times a day, 5 days a week for 4 weeks. And so you can imagine, to really develop the disease, you need to smoke these mice for 6 months. And normally, the wild-type mice have increased neutrophils, just like you see in a patient. But the glutaredoxin mice, when they receive smoke for 4 weeks, they do not develop as much bowel neutrophilia. Also, the main chemoattractant for the neutrophils is not induced as much by the cigarette smoke in the glutaredoxin knockout mice. So this very important antioxidant enzyme, which we've shown in vitro, was connected to inflammation and NF-kappa B signaling. Also in this mouse model shows a protective effect. So less glutaredoxin, less NF-kappa B, less inflammation, just like we showed in the cell culture system. We're of course interested in the patients with COPD, not mice with a COPD-like phenotype. So can you imagine how you get material from patients, lung material from patients, to investigate a question like this?
Sputum samples.
Yeah, I guess it's on the slides. And do you have any idea what that means?
It's mucus that patients cough up, and it has a lot of leftovers of destroyed cells, etc. and cytokines.
And so if we also need a control healthy population to compare it to, how would you get the same type of material?
I think they give them certain cocktail which makes the healthy patient use mucus as well.
Mm-hmm.
And then they
So would anybody volunteer to inhale? It's really just a saline solution, so just a salt solution that people inhale, and then they start naturally coughing up mucus. Does it sound pleasant to anybody? Most volunteers, of course, get reimbursed for any damage or discomfort for their time. But as part of the problems with lung research, I mean, you can take material that comes from the operating theater where patients get lung cancer resections, and you take healthy tissue that is resected as well. But there's always a risk that what you're looking at is somewhat cancer-derived and not necessarily COPD-derived. So sputum is an in-between method. It does include damaged cells. It contains a lot of the inflammatory cells, but it doesn't really contain healthy epithelial cells, which are the main cell of interest in our research. But it's as close as a not very invasive method of obtaining some indication of what's going on in the lung. So we received some of these mucus samples through a collaboration with the University of Liège, where they do this more in a routine manner than we do here in Maastricht. So we had healthy non-smoking controls, healthy smokers, because you have to also look at whether there's an effect of smoking by itself or whether it's really a disease process. We had stable COPD patients and patients that were going through an exacerbation. So those are typically patients that are having a cold, a viral infection that is worsening their symptoms worse than they normally are. So we looked in those sputum samples for the gluriodoxin 1 protein by Western blot. And so we did see that smoking somewhat increases the amount of gluriodoxin that is being coughed up, so to speak, but that it's mainly the patients that are experiencing exacerbation that have increased gluriodoxin 1 levels. Now, whether that is an active secretion mechanism or that's part of the damage that is being caused, that we don't know yet. We also looked at how much oxidation of glutathione to proteins was occurring, and here we see the inverse. Patients with a COPD disease exacerbation have decreased levels. So it nicely fits because more gluriodoxin would take more glutathione off of proteins. So there was a nice relation. But this is purely observational. We do not know at this point what it means to have been coughing up more gluriodoxin, less of this oxidation. One way in clinical samples to try to find associations is to look at a correlation. So we looked at correlations with lung function, and these are some of the lung function characteristics. FEV1% predicted, which is how much air a patient can exhale in the first seconds. I told you the patients really have a problem exhaling the air, and a normal, healthy being will exhale close to 100% of all its exhalation capacity in that first second. But in a COPD patient, this goes down, depending on the disease state, it can go down to 70%, some people even only 20% of what a normal person can exhale. They exhale in the first second. So it's a measure of disease severity. So we plotted, this is just an artificial plot to make you understand correlations. We found a positive correlation between the disease severity, lung function, and the amount of glutathione bound to proteins. So the better the lung function, the more protein bound glutathione they are coughing up. So also indicating that it's somewhat protected. The gluriodoxin 1 levels, they show the inverse trend. So the better the lung function, the more gluriodoxin, the less gluriodoxin, was being coughed up. So it does indicate that there is a relation between these oxidations, this antioxidant, and lung function. We also were interested, of course, in inflammation. So in the sputum, you can not only look at cells, but also some of the soluble mediators. Two important cytokines, again, IL-8 as well as IL-1 beta. And so, again, a negative correlation here between the inflammatory markers and the amount of the oxidation. And for gluriodoxin, just like with lung function, the inverse. So less gluriodoxin means less inflammation. And this is exactly what we found in vitro as well as in the mouse model. Are there any questions on this part of the presentation? So I've given you an example of oxidants working as signaling molecules, but on the other hand also an antioxidant being involved in regulating lung function and inflammation. I want to give you a second example of the more classical damage, but damage that can also act as a signaling molecule. And the example I wanted to show was advanced glycation end products, or AGEs. Now, AGEs are formed through the oxidation as well as the utilization of glucose and lipids. It's very complicated chemistry, lots of chemical terms, but it's just important to know that lipids and glucose, like in diabetics, there is a higher level. This will lead to higher level of oxidation, formation of very toxic intermediate products, and they can ultimately couple to proteins. And then again, of course, they can affect function of the proteins, but what these specifically also do is activate a receptor, namely the receptor for advanced glycation end products. So you have sugars that are coupled to proteins, and therefore it's called advanced glycation end products. So the receptor for advanced glycation end products is called RAGE, and binding of AGEs to RAGE will lead also to the activation of NF-kappa B, so perpetuating inflammation, but it can also enhance even more oxidant production. So there's sort of a feed-forward mechanism. The AGEs are formed because there's oxidative stress. The AGEs bind the receptor, and that can induce more oxidant production. For RAGE, there is, so normally the membrane-bound receptor that can signal, but there's also different isoforms of soluble RAGE which do not sit in the membrane and they cannot signal. So it's thought that they are actually anti-signaling molecules. AGEs are known to induce NF-kappa B. This is an example of where they treated cells with either native BSA, which is in lane 3, so just BSA-treated cells, but then they also oxidized the BSA with a sugar, CML-associated BSA in lane 2. And you see that if you look at the NF-kappa B activity, this is binding to DNA, that adding the BSA that is oxidized induces more NF-kappa B activity than when you just do an unmodified BSA treatment of the cells. Comparing lane 2 and lane 3. And in lane 5, what they did to show that this was dependent on the receptor, they added an antibody to the receptor. Now, the band intensity isn't much difference, but if you compare lane 5 to lane 2, there is somewhat an attenuation of NF-kappa B activity if you add this anti-RAGE antibody. And also in lane 6, they added excess of the soluble receptor. So if you have the soluble receptor competing for the membrane-bound receptor, there is less activation of the NF-kappa B signaling. On the other side of the slide, they also looked at when lungs, so whole animals, and they were treated with BSA that was either non-modified in lane 1, or they modified the BSA with an oxidation of sugar. So again, CML BSA in lane 2. And in the lung tissue, this led to an upregulation of a main adhesion molecule for inflammation, namely VCAM. And they just use LPS here as a positive control, so a known inflammatory trigger. AGEs are, for instance, also known to induce proliferation of macrophages. So again, one of these feed-forward loops. You have oxidative stress mainly derived from inflammation. You get AGE formation, and the AGEs can signal further to more macrophage proliferation. So the open circles are the BSA that is modified by oxidized glucose, and the dark circles are unmodified BSA. So if you modify the BSA, you get more proliferation of macrophages. We also looked in our COPD patients, and this is pentosodine, so one of the advanced glycation end products. And we did this in the plasma of the patients. So it's not only diabetics who have increased levels of glucose oxidation, it's also other chronic inflammatory diseases such as COPD. So the COPD patients in their plasma, compared to the controls, have a higher level of these advanced glycation end products. And we saw this both for pentosodine as well as carboxyethyl lysine, so specifically modified lysine proteins. There wasn't as much of an effect of disease severity, so we classify COPD into the GOLD categories. So the higher the GOLD number, the worse the disease is. But you see an increase of the advanced glycation end products in COPD in general, but it doesn't seem to be very associated with the lung function. What is also very interesting about advanced glycation end products is it's an irreversible oxidation, and so you mainly will detect it on proteins that do not have a rapid turnover. And one of those proteins with a very low turnover rate is actually the collagen in your skin. And collagen contains a lot of lysine residues which are targeted by this oxidation. So people in Groningen, they actually developed sort of a luminometer. So people just have to put their arm on this machine, and because of the light that is being shone on the skin and the light that is being reflected, you have an idea of how much of this oxidation of collagen is occurring in the skin. So we were also interested in finding out whether this would be a non-invasive way of looking at how bad oxidative stress is in COPD patients. So you don't have to collect blood, you don't have to puncture a vein, you don't have to have them cough up sputum or go to a lung sample. Can we just put their arm on a luminometer and see if we have an idea about the oxidative stress? So here the oxidation of collagen in the skin. And this is a normally occurring phenomenon. So if you take young people versus old people, you see this accumulation increasing. But the thought that is in COPD, this is accelerated because, of course, smoking, which causes a lot of oxidative damage, but also because of inflammation. So we looked at the fluorescence from the skin, so measure of the oxidation of the collagen, and we found that this was markedly increased in the COPD patients compared to the controls. But again, it didn't seem to really depend on how severe their lung function was affected. The receptor for these advanced glycation end products, before birth, the lung has no expression whatsoever. And once babies or mouse pups are born, the second day, the RAGE level is the highest of the entire body. So there must be some really important role for this receptor in the lung. And in COPD, I told you that the alveolar spaces are destructed. So we expect that there will be less expression of RAGE because there's just less cells to express it. So a paper from last year actually showed that this is another technique. It's not coughing up sputum. You actually have to go with a bronchoscope into the lung under mild sedation of the patient. And you rinse a part of the lung with saline solution, and then you can measure metabolites in this lavage fluid. So what these people show that if you compare the lavage fluid of healthy controls versus COPD patients, there is indeed less of this RAGE, so this H receptor present. Then we also looked in the plasma to see if the levels in the lung reflect the level in the plasma. And in this paper, it is shown that if people have more emphysema, so more destruction of the lung tissue, less cells expressing RAGE, that there is also less of this RAGE receptor present in the plasma. So I hope by giving these two examples of oxidants as signaling molecules, either really signaling or damaging signaling, that they are more than just damage markers in disease, which they've been used for decades in the past. So they really play an active role in disease processes, and therefore they can also be targeted more specifically than with general antioxidant approaches, because every single disease characterized by chronic inflammation and high levels of oxidative stress, people have tried to treat with general antioxidants, but that has rarely resulted in positive results. And so if you look at these examples, it might be that you're not just restoring the balance, but you're also tipping the balance towards too much antioxidant levels, especially in healthy people. If you don't have a problem with antioxidant levels, then there is no reason to try to fix it. And any antioxidants can also work in a different way, in a damaging way. So I think there is still value in using antioxidants for treatment of disease, but if you understand the signaling processes these oxidants are involved in better, then you can also target and use more specific antioxidant therapies than what have been used so far. So that was what I wanted to tell you today about our research in lung disease and redox signaling. Are there any more questions or